Hey everyone, I'm Dr. Fizz and let's learn some physiology. We're skipping our intro again today for this video series because I do not have enough time to edit this. Alright, so we're going into smooth muscle and let's get right into it. So smooth muscle is super important for our system. It's found within our system for the most part, but we're going to talk about three different subjects with this. We're going to talk about what it is and what types of smooth muscle exist. We're going to talk about the types of smooth muscle contraction, and we'll also talk about how skeletal muscle or smooth muscle compares and contrasts. All right, so comparing smooth muscle to itself, really, smooth muscle is found all along the hollow organs of the digestive system, the airways of our lungs, like our esophagus and whatnot, tubes of our urinary system, and our blood vessels even. So it's a lot slower than skeletal muscle, and we'll talk more about that later. But it generates a lot more force per area, and it can shorten about 80% of the length of its actin to myosin, which is pretty dramatic. We'll talk about the difference between skeletal muscle and smooth muscle there. It's super fatigue resistant, and we're going to talk about why. All right, so smooth muscle is split into two groups. We'll talk about its subtypes. The first unit here, let me show you with my laser pointer, my trusty laser pointer. This is called single unit smooth muscle, and this is called multi-unit smooth muscle. So single unit smooth muscle are connected by something called gap junctions. You'll have a lot of smooth muscles that are connected together, and what will happen is an impulse was sent down to one cell. That cell will then propagate its electrical signal throughout the gap junctions, triggering other cells to react, and then send it back out of the nerve cell if it needs to, or receive another signal in a different direction. And then send waves back and forth. That's really what it looks like. These act as waves. For an example, in the GI tract, we find something called peristalsis, I believe, and that acts as a wave going through the GI tract to help move our system forward. Let's see. They allow for coordinated contraction, which ends up being more powerful. Things like the pacemaker acts with gap junctions between the smooth muscle cells, and therefore these can be independent of the nervous system, just because flow of ions in the channel can cause chain reactions, and it doesn't exactly need a signal from neurons to even start. So that's what's kind of cool about this. And then moving on to multi-unit smooth muscle, we have these isolated cells here. You can see that there's no gap junctions. They're all innervated by nerves. These allow for fine motor control. Imagine if you're trying to really just twist something ever so slightly, you're going to need a lot of cells acting together in different directions in order to really get that task done. So those are super helpful. They rely solely on endocrine. I guess it was a bad example because that was skeletal muscle, but you get the idea. They rely on endocrine for hormones, and we'll see that in just a bit, or even the nervous system, as we can see from this picture. All right, moving on. So smooth muscle contraction is important. Our skeletal muscles contract, our smooth muscles also contract, but they contract in different ways. Although there are a lot of similarities, as you'll see here, the cycle looks pretty similar. There are some things that are different in here, so let's talk about them. We still have our myosin, we still have our actin, but our actin does not have, or F-actin here does not have troponin found along the G-actin molecules. So you have tropomyosin still. You can see the purple within these red circles. It might be a little hard to see, but there is no troponin. So that begs the question, if troponin was the thing that reacted with calcium to allow tropomyosin to move and expose the active sites, then how does this myosin act like a myosin then? Well, let's talk about that. In smooth muscle contraction cells, we have something called calmodulin, which is found within the system. Calmodulin is the thing that reacts with calcium. That undergoes a conformational shift, then combined with something called myosin light chain kinase, or MLCK, will undergo another conformational shift, which exposes the active site of MLCK, which attaches to myosin, which allows it to bind to G-actin. So that is that. The question is, where does this calcium come from? It comes from extracellular influence and intracellular influence, just like skeletal muscle. Well, for skeletal muscle, it was a lot more sarcoplasmic reticulum, but for this one, we'll get a lot of calcium from the extracellular fluid or storages as well. So that extracellular storage is activated by different membrane potentials. So if ions were to leave a channel, it open up a gate, open up a storage system on the outside of the cell, and allow for calcium to flow into the cell, thus triggering calmodulin. We talked about endocrine system earlier. This is where it comes into play. Hormones or different molecules can attach to a G protein, which will send an alpha subunit on this G protein, the end of the unit right there, to hit phospholipase C. Phospholipase C will then trigger and send out IP3, which is a signaling molecule to activate a calcium channel on the sarcoplasmic reticulum. I don't know if it's RYR or not. It might be, but that allows for calcium to leave the channel, and from there, circuit pumps pump back calcium into the cell when necessary. All right, so moving on, we have just like our skeletal muscle system, we will have ADP and PI leave, but then we have that blue phosphate that's still on here from the MLCK is attached. ATP will come in, pop it off, and something called myosin light chain phosphatase will actually cleave that phosphate group, causing it to go back into a relaxation phase and go back to the first step. So this cross bridge right here says if this blue stays on, if there's not enough MLCP to cleave that, it'll just keep going around and around in the cycle until there is, and then it'll go back into the relaxation phase. Something that can cause that phosphatate myosin light chain MLCP not to be active is if rho kinase was activated, which throws on a phosphate group and inhibits our myosin light chain phosphate. So poor little Pac-Man. All right, so smooth muscle contraction. That last one was called phasic. It was short, just and can allow for short contractions like skeletal muscle. Smooth muscle contraction in a tonic sense is a lot longer. It goes through the same process, but say we were to have more MLCP in the system than we were to have MLCK, then our system would have MLCP all over the place, and it would more than likely clip off that phosphate prematurely, thus leaving it with a naked myosin there, and causing it to detach. But the hydrolysis state wouldn't happen because all the phosphatases were over here, and occupying all this area over here, where there would be a longer wait time to be phosphorylated over here, and that blue phosphate already been clipped off here wouldn't allow this to transfer from this stage to this stage as fast. This is called a latch state, and it happens in a tonic muscle like a sphincter. All right, so let's compare it to skeletal. Let's compare skeletal muscle to smooth muscle real quick. I said that smooth muscle can contract, shorten to about 80% of the length of the myosin and I guess the sarcomere length. Skeletal muscle only shortens 30%. So, if you think this is impressive, you should check out the GI tract. It's pretty efficient. So there is a difference in myofilament structure there, and smooth muscle is 10,000 times longer than the fibers in skeletal muscle, and they have they still have sarcometric arrangements. So, all right, let's see. Comparing other things with this. Smooth muscle does not have troponin, like we mentioned earlier. It does not have T-tubules either. So there are no invaginations on these like skeletal muscle will have. And so calcium does not move troponin in the same in any way really, but skeletal muscles, calcium is geared to move troponin um, through conformational shifts. So tropomyosin through conformational shift of troponin. So that's also super important. The skeletal muscle is kind of deemed as a thin chain or a thin filament regulated molecule. Like the actin molecule is really what makes it up. But smooth muscle is it all has to depend on the thick filament, which is the myosin. This is why it's called heavy chain regulated. That's about it. But in my comparison between the two, but we know that we mentioned the entire time that there are a lot of differences here. And we can see that these cells are single cells and they're singularly nucleated, where if you were to look at this, there would be a lot of nucleus around each fiber or cell on the skeletal muscle. Anyways, so that has been this video for today. If you have any questions or if you need me to answer more things, I'd be happy to. Just reach out to me. And that's about it. So stay busy.